Hello friend, in this module we are going to see the complete flow of events for the bioequivalence study. So the flow of events right from the start to end will be presented in this particular module. Before going into the flow of events, here are a couple of definitions as per the good clinical practices. A sponsor. In the conduct of clinical trial, a sponsor is an individual, institution, company or organization that takes the responsibility to initiate, manage, finance the clinical trial, but it does not actually conduct the investigation. So that company or organization called as sponsor. Then contract research organization. A person or an organization, it can be commercial, academic or other, contracted by the sponsor to perform one or more of a sponsored trial related duties and function. We can classify the flow of events of a bioequivalence study or a bioavailability study into three major divisions. One is the pre-study events, then the events during the study, and the post-study events. So we will start with the pre-study events. So any kind of activity happening in a CRO, there should be a receipt of study confirmation from the sponsor. So he is the authority who is going to sponsor the entire activities, who is going to pay for that. So there should be a confirmation or a contract execution between the sponsor and CRO should happen before the study initiation. So that once the contract is signed, then the study initiation will be happen. Then the study plan will be prepared tentatively by the entire study team. Then the study design will be carried out by the pharmacokinetics and statistical team along with the investigators and analytical investigator as well as the clinical investigator. Then once the study design is finalized along with the sponsor, the protocol development will start by the protocol development department. Then the protocol will be finalized in agreement with the sponsor and the internal stakeholders. Then finally, the protocol will be signed off by approval from the sponsor. Then the protocol related documents like informed consent document, case report forms, meal menu, diet plan, etc. will be finalized. Further, the pre-study events continues like ethics committee dossier preparation. So once the protocol is ready with along with the protocol related documents, the ethics committee dossier with along with the application will be prepared. Then it will be submitted to the ethics committee. Then the approval of the protocol from the ethics committee will be obtained. Then the entire package along with the protocol, along with the ethics committee, then the formulation details, whatever required for the regulatory submission, that is nothing but the CDSEO online submission. The entire dossier will be prepared and that will be submitted through Sugam portal of the CDSEO. Then the NOC application will be tracked by the CRO for further queries or any other clarifications from the CDSEO. Once the queries and clarification is addressed by the CDSEO, then the NOC will be issued by CDSEO for the particular study. Then in case import license is required for the import of investigational products, then the import license also parallelly applied along with the NOC application. Then both the NOC and import license will be obtained. If the investigational products need to be imported from outside India, then the receipt of investigational products will be obtained by import from the foreign country. Pre-study events also comprises the bioanalytical method development. Once we come to know about the drug product and drug, the standards for the particular drug will be obtained and the bioanalytical method development will be started. So the development and validation of the bioanalytical method is important before starting the study because how to handle the blood samples, what is the processing procedure as well as the storage conditions will be defined by the bioanalytical method. For example, any stabilizing agent, any specific temperature need to be confirmed for the particular drug product, then the bioanalytical method should be in place. So before starting the clinical phase of the study, the readiness of the bioanalytical method is very critical and the sample handling procedure also very critical for before starting the clinical phase of the bioequivalence study. Then the study information advertisement for the study volunteers also will be issued in case the volunteers are not sufficiently obtained by the volunteer recruitment officer. So once the bioanalytical method is ready and the volunteers are ready, actual schedule of the study will be arranged. Now, the actual study event starts the clinical phase of the study. So first the screening of the volunteers will happen. So it is a healthy subject screening. So the blood sample will be collected for to prove the healthiness of the subject. Along with the blood sample, ECG will be taken, urine testing will be taken, physical examination will be done, X-ray will be taken. So all these testing will be carried out to understand whether the volunteer is healthy or not. Once it is investigated by the physician, then he will be given the fitness for the study. So before doing this screening, the volunteers will be asked to provide the informed consent for the screening so that the volunteers will be knowing what kind of test they are undergoing, and there should not be any concern from the volunteer side. So for that, the consent will be obtained. So once the fitness for the volunteers will be obtained, then the selection of subject based on the inclusion exclusion criteria in provided the protocol will happen. Then the study specific informed consent procedure will be carried out for the selected subjects for the study. Once the informed consent for the study will be obtained, then the volunteers will be checked in into the housing facility of the bioequivalence center. So during the check-in, the subject will be checked for alcohol presence in their blood or drug of abuse in the blood. If anything that sort is positive for the specific volunteer, they will be excluded from the study. So in general, the check-in of the subjects will happen 12 to 15 hours before the dosing of the investigational products. So if the dosing is scheduled today morning 7 o'clock, then check-in of the volunteers one day before. For example, if it is Tuesday 7 o'clock, I schedule the dosing. Then Monday evening, before 7 o'clock, the subject will be checked into the facility. For the overnight fasting, if it is required, overnight fasting will be carried out. It is minimum 8 to 10 hours, depends on the regulatory agency. So the volunteers will be provided dinner after the check-in. Then they will be fasting for at least 8 to 10 hours before the dosing. So during the dosing day, the volunteers will get up, then they will be ready for the study activities. So the pre-dose sample first will be collected from the volunteers, then the dosing of the investigational product will happen. So during the dosing, in case of fasting study, directly the dosing will happen. In case of fast study, the breakfast will be provided, whether it is a high fat, high calorie breakfast or a standard breakfast, depends on the protocol, it will be provided. Immediately after completion of the breakfast, the subject will be dosed for the investigational product. So after dosing, there will be water restriction for at least 1 to 2 hours so that there won't be any external factor influence on the drug absorption. Then after immediately dosing, the blood samples will be collected as per the time points. So the study events continue to be happen. The collected blood sample will be processed and stored in the clinical facility of the bioequivalence center. The samples will be processed and stored as per the sample handling procedure. Then the safety assessment as per the protocol driven time points will happen. The vitals will be monitored. In case ECG need to be taken, the ECG will be taken in between during the study also. Then after 4 hours from the breakfast or after 4 hours from the dosing, the lunch will be provided. And then after 4 hours, again 3 or 4 hours, the snacks will be provided. Then after again 3 to 4 hours, dinner will be provided for throughout the dosing day as per the meal menu. So generally, the housing of the subjects will happen 24 hours after the dosing in most of the studies, and sometimes it depends on the protocol, 48 hours, 72 hours also the subject will be housed based on the protocol. And finally, the checkout will happen once the study samples have been collected. Sometimes the study samples has to be collected by ambulatory collections, like the subjects will go out and come for every 12 hours or 24 hours to give the samples at the facility. That is called ambulatory sample collection. That will happen. Like this, the another period of the clinical phase also will be completed. Second period, when the subjects comes, again the check-in formalities will be completed, like alcohol abuse and drugs of abuse test will be carried out, physical examination, or if required, some blood sampling if required. Most of the time it is not required, but during the short washout period. But sometimes if any other things need to be confirmed, that test also will be done. So the subject will get completed both the periods in a similar way. Then finally, all the samples collected, blood samples will be collected, will be transferred to the bioanalytical facility along with the clinical summary, whatever any adverse events happen, any concomitant medication is given along with the, for example, we are doing study for paracetamol and you have given something else like omeprazole or something for the acidity or something. So that also will be reported for the in case any interference in the bioanalytical stage, that also will be checked. So that will be mentioned in the clinical summary. Any dropout details, any withdrawn subject withdrawal details, all those informations will be there in the clinical summary. After the shipment of sample from the clinical facility to the bioanalytical facility, the bioanalytical lab in charge will receive all the samples all with all the clinical information. Then any information which is related to incomplete withdrawn subjects, vomiting details, concomitant medication, subjects to be analyzed as per the protocol will be noted down by the bioanalytical investigator. Then the initiation of subject sample analysis will happen. So all the subject sample will be analyzed sequentially based on the analytical plan. Then once the all the subjects analysis happen, if in case any analytical run failure happens, the repeat analysis also happens. Then incurred sample reanalysis. Some selected samples will be reanalyzed to check the integrity of the bioanalytical method. It is a USFDA requirement, not in other regulatory agencies. Then once the all the analysis is completed, the entire bioanalytical data, generated data will be quality control procedure will be applied. Then the quality assurance procedure will be applied. Then the final data will be transferred to the pharmacokinetic analysis into the pharmacokinetic department where the analysis will happen. Pharmacokinetic analysis through software will happen, and along with that, the bioanalytical report will be prepared with all the method validation development report and the subject concentration data. From the bioanalytical department, the QA'd time point concentration data will be received by the PK analyst. Then the PK analyst will prepare the input data for the PK calculation. So input data in some format, some specified format will be prepared in case if it is for the Winolin kind of software, the specific format will be prepared. The input data generally contains the time point, the concentration at specific time point, subject number, period number, sequence, and if any other information, if there any change in the dose or something. So those all information, critical all information will be incorporated in the input data. Then the input data will be taken printout and it will be QC QA'd for any wrong entry or anything, any changes in the data that will be checked and the correct data will be ensured for the input. Then finally, the input data will be taken by the PK analyst to calculate the PK results by the Winolin or other applicable software. Then the review of PK data, the output of the data will be reviewed by the PK analyst for any abnormal data or anomalies or the first time point CMAX if anything is there. So any other things, any abnormal curves are there, this all will be reviewed by the PK analyst. Then finally, based on the PK output, PK data will be summarized to prepare the input data for the statistical analysis for the conclusion of the bioequivalence. The input data again need not to be checked because it will be the output from the software. So that will be arranged as per the requirement and randomly reviewed. The input data like Cmax for every subject or and AUC and any other primary parameter, all the parameter will be taken for the bioequivalence analysis. The execution of bioequivalence statistical analysis will be carried out by either Winolin or SAS software. Then finally, the summary of results, summary statistics, all the plots, linear plots and semi-log plots will be provided for the report preparation. Finally, the report phase of the bioequivalence study will happen once all the analysis has completed. So the report person will collect all the information right from the clinical data, summary, clinical report, or the bioanalytical data and reports, PK stat data and reports, graphical representations like plots, linear plots and semi-log plots. Everything will be collected by the report preparation officer. Then the report will be prepared as per the ICH E3 format provided by the regulatory agency. Then the investigators and the quality control department will review the report. Then the quality assurance department will review the report. After that, the report will be sent to the sponsor for their review. Then finally, after all the review comments will be incorporated in the report and the report will be finalized along with the appendices like protocol copies and ICD copies. So many appendices as per the E3 format will be required. So all these things will be finalized. Then once the report is finalized, it will be converted into PDF format. Then the PDF will be bookmarked, hyperlinked. The tables and the figures will be hyperlinked to the table of contents. Though this process is called as generally ECTD formatting. So the PDF copies will be having for the easy review of the reviewers, whatever possible in the PDF like interlinking and bookmarking, these things will be carried out by the report preparation personnel. Then finally, this report will be submitted to sponsor for the further submission to the regulatory agency. Finally, the documents related to the study will be archived as per the CRO procedure. So with this, the end of all the sequence of events for the bioequivalence study gets completed. In this module, we have seen the flow of events for each and every phase of the bioequivalence study, and in the upcoming module, we will see in detailed manner the study designs, different study designs adopted for the bioequivalence studies. Thank you.